Hello and welcome to my presentation. Superovulation with different trigger agents differentially alters leukocyte expression in the peri-implantation endometrium in a mouse model. My name is Valerie Libby. There are no conflicts of interest, although this research was funded by a Prelude Scientific Research grant. The majority of children born following assisted reproductive technology are healthy. However, compared to spontaneously conceived children, singleton gestations after IVF are associated with increased risks of adverse obstetric and perinatal outcomes, including hypertensive disorders of pregnancy, placentation disorders, low birth weight, small for gestational age, and preterm delivery. Interestingly, the use of frozen embryo transfer into a non-superovulated endometrium decreases many of these risks when compared to fresh embryo transfers. Superovulation, the administration of exogenous gonadotropins for multifollicular recruitment, followed by an LH analog or trigger agent to mimic ovulation, results in supraphysiologic levels of hormones which can have significant impact on the endometrium. In particular, studies have found altered endometrial angiogenesis and vasculogenesis after superovulation, which may contribute to the mechanism of impaired implantation and abnormal placentation observed in superovulation. Our mouse model of IVF recently demonstrated that superovulation, as well as the choice of trigger agent, significantly impacts endometrial VEGF bioavailability and subsequent angiogenesis. Trigger agents have different effects on vasculogenesis, endometrial receptivity, and pregnancy outcomes. The two ovulation trigger agents we use to mimic the LH surge in IVF are HCG or Lupron, a GnRH agonist. The HCG trigger with a longer half-life results in increased stimulation of the corpus luteum and subsequent increased ovarian VEGF production and endometrial VEGF receptors, which increases the risk for OHSS. Lupron, on the other hand, has a shorter half-life, resulting in a possible luteal defect, but has the advantage of decreased VEGF production and decreased risks of OHSS. However, a recent Cochrane review found that Lupron also results in decreased implantation, pregnancy, and live birth rates and increased risk of miscarriage. The mechanism at the level of the uterus behind which these trigger agents have differential effects is not well known. It is well known, however, that the endometrial immune environment is changed after superovulation. Specifically, special cells called uterine NK cells, which make up to 70% of our decidualized endometrium, have been found to play a key role in early pregnancy and placentation. Studies have found decreased levels of uterine NK cells after superovulation in both women and mice, as well as in endometrial biopsies of women with recurrent implantation failure. Many of these studies have been cited when discussing the benefits of frozen embryo transfer over fresh embryo transfer. There is no data on the effects of the trigger agents on uterine NK cells during early pregnancy. This is the first study to look at the effect of the GnRH agonist versus HCG trigger on immunologic processes in the murine preimplantation uterine environment. Our objective is to determine the impact of superovulation with final oocyte maturation induced with HCG or Lupron on leukocyte and uterine NK cell populations in murine gestation during preimplantation. We hypothesize that the GnRH trigger differentially alters leukocyte expression, contributing to the aberrant placental development and function, which may have clinical implications in human IVF. Now I will present the methods. Female mice were assigned to one of three protocols. Group 1 was the control group, naturally mated without superovulation or trigger. In groups 2 and 3, female mice were superovulated with PMSG followed 48 hours later by either an intraperitoneal injection of HCG trigger as in group 2 or a Lupron trigger as in group 3. All mice were mated to fertile males, which was verified by checking vaginal plugs the following day. Females were then killed prior to implantation and maternal uteri and ovaries were collected. The uterus was divided at the midline into two separate whole pieces of tissue. One ovary and half uterus from each mouse was fixed overnight for histologic analysis, and the remaining whole mouse uterus from each mouse was snap frozen and stored at -80 degrees for PCR. Total RNA was extracted, prepared, and reverse transcribed from frozen mouse tissue. This was followed by quantitative real-time PCR on each sample for relative mRNA expression of immune cells, including leukocytes and uterine natural killer cells. Ovaries and remaining half uteri were fixed, dehydrated, and paraffin embedded. Serial tissue sections were cut and mounted on glass slides. Immunohistochemistry was performed with staining for antibodies to CD45, the common leukocyte antigen, to localize the expression of leukocytes, and CD31 to localize angiogenic markers. Tissue H&E staining of uteri was performed to assess endometrial gland density, and H&E staining of ovaries assessed response to superovulation by quantification of corpora lutea. A priori power calculation demonstrated that 5 mice per group would provide 80% power at an alpha of 0.05 to detect an 8% difference in uterine NK cell expression, a number that was based on our lab's preliminary data. All statistical analyses were performed using GraphPad Prism, and one-way ANOVA followed by post-hoc 2Ks multiple comparisons tests were used to assess differences in gene expression between the 3 groups. Differences were considered statistically significant when p was less than or equal to 0.05. Now I will present our results. In order to confirm that our model of superovulation was successful, we examined the ovaries of control mice to mice superovulated with PMSG followed by one of the triggers. Here you can see an ovary of a representative control mouse with the corpora lutea, an ovary of a mouse that was superovulated and triggered with HCG, and finally a mouse superovulated and triggered with Lupron. We found significantly more corpora lutea in both superovulated groups compared to the non-superovulated control group. There was no significant difference between the 2 superovulated groups. This confirms that our superovulation worked with regards to ovarian response. Now I will present the results from immunohistochemistry and H&E staining. To assess the impact of trigger agent on leukocyte density, we used histopathology with H&E and immunohistochemistry. Superovulated mice had significantly decreased uterine protein expression of leukocytes compared to naturally mated controls, with HCG-triggered mice showing significantly increased expression of uterine leukocytes compared to the Lupron group. Leukocyte density per gland was significantly increased in the HCG trigger group compared to both the Lupron and the non-superovulated groups. There was no difference in leukocyte density per gland between the controls and the Lupron trigger groups. To investigate the relationship between changes in endometrial leukocytes and angiogenesis, we examined vessel density by CD31 protein expression on immunohistochemistry and gland density from H&E histopathology of preimplantation uteri. Compared to controls, superovulation resulted in significantly decreased uterine vessel density. There was no difference in vessel density between the trigger groups. Similarly, both superovulated groups demonstrated a trend of decreased vessel density per gland when compared to controls, with no difference between trigger groups, though this result was not significant. There was no significant difference in gland density between groups, but there was a trend towards decreased glandular density after superovulation, particularly after the HCG trigger. Now I will present the results from PCR. PCR was performed to assess mRNA expression of key endometrial leukocytes using CD45 as a general white blood cell marker and specific uterine NK cell markers. There was no significant increase in white blood cells or uterine NK cells mRNA expression between all 3 groups. However, the Lupron-triggered group had a trend of more white blood cells and uterine NK cells compared to the HCG and control groups. In contrast, the HCG trigger group appeared to have the lowest amount of uterine NK cells. The N was low in these groups, which may limit the power of interpretation. This was the first mouse model with Lupron and HCG triggers to investigate immune markers of early pregnancy. We did find differences in leukocyte expression and vessel density and trends in uterine NK cell expression after superovulation and between trigger agents. These findings could form the basis for future investigation into birth outcomes after superovulation and Lupron triggers. There are a few small case series on outcomes of human live births following Lupron trigger, and the effects of Lupron on birth outcomes are unknown. This study was limited by the fact that it was a mouse model. We used only pre-implantation time points, and this is our preliminary study, and future research using a murine embryo transfer model will be useful to understand later pregnancy outcomes. In conclusion, this is the first study to delineate the effect of superovulation with different triggers on markers of endometrial leukocytes in a mouse model. The triggers differentially disrupt endometrial expression of leukocytes and may differentially disrupt uterine NK cell expression. Future studies could investigate the downstream effects of superovulation with the HCG and Lupron trigger on leukocyte expression, placental vasculogenesis, and pregnancy outcomes using an embryo transfer mouse model. The implications may be significant for placental formation and neonatal outcomes following human IVF. Thank you to everyone that helped me with this study, including Dr. Wienerman and Messiano. These are my references. Do you have any questions?